Our next speaker, Killian Hanlon, is a research fellow at the Harvard Medical School at the Massachusetts General Hospital.
Hi everyone, thanks for coming to my talk and thank you to the organizers for inviting me. So today I'm going to talk to you about some AAV variants we've created and how we've translated them, how we've taken them from the mouse to the macaque in the context of the spinal cord and the inner ear. So first off, these are just my disclosures for this talk. I'm sure most of you are familiar with AAV. Many AAV libraries use random peptide insertions in the regions highlighted in purple in the above image. In this method, each variant carries its own capsid gene containing the insert sequence, which conveniently also acts as a barcode. The standard approach is to inject a cocktail containing millions of these variants into a tissue of interest and go through rounds of selection to isolate a superior variant. However, a common issue with this is that the variants you get out at the end are often non-functional. There are many reasons for why this is, but we believe it's partly because AAV transduction is a multi-step process. First you have to get to the target organ, then you have to bind to the right cell, you have to get internalized, get to the nucleus, and finally finish uncoding and drive transgene expression. And most selection schemes don't differentiate between these first few steps when really selecting at this final step for vectors actually capable of eliciting transduction is the ideal strategy. And this is the approach we've taken. As well as carrying the cap gene, our variants also incorporate a Cree expression cassette. This allows us to inject our library into a transgenic mouse carrying a Floxed-STOP fluorescent reporter. Variants that express Cree will unlock expression of tdTomato in this case. This allows us to flow sort out positive cells, co-sorting with a cell marker of interest, with the hope that variants purified from these cells will show enhanced properties. So our workflow then becomes injecting the mouse, pulling out cells from our tissue of interest, amplifying the viral DNA. We use low-depth NGS just to check our levels of enrichment, and if need be, recloning the variant inserts and performing another round of selection. So as a test of this, we wanted to see if we could develop a variant that could transduce the brain and the CNS intravenously more efficiently than the parent capsid AAV9. And to do this, we injected our Floxed-STOP tdTomato mice with our library. We harvest the brain tissue 3 weeks later, and we perform flow cytometry on that. After a first round of traditional library selection, just to weed out dead capsids, we performed one round of Cree-based selection. Coming out of that, more than 3/4 of sequences consisted of just 2 variants, which we called AAVF and AAVS. So to test these, we produced vector of each carrying a CBA-GFP construct and tested this intravenously in C57 mice. We compared expression of AAVF and S to both AAV9, the parent capsid, as well as PHBB, a well-known variant developed using an advanced library system that expresses much better than AAV9. While AAVS failed to express better than AAV9 in the brain, AAVF drove extremely robust, high-level expression across the CNS. This expression was consistent between mice of different sex as well as different strains of mice. We were surprised at the inability of AAVS to cross the blood-brain barrier, but nevertheless we saw high peripheral expression as well as high expression in the brain when injected locally, which indicated the capsid was functioning. Because AAVF looked so great in the mouse, we wanted to see if this same pattern would be reflected in larger animals, such as non-human primates. So we performed a pilot 3-week study in a small number of animals, just injected with either AAV9 or F, both carrying GFP. And animals were injected with a relatively low dose of 3 to 4 by 10 to the 12 vector genomes. We found that AAVF expresses very well in the non-human primate spinal cord, transducing neurons as well as astrocytes, along with small numbers of microglia and oligodendrocytes. The bulk of expression was seen in interneurons and motor neurons that you can see here, and as the images reflect for both AAV9 and AAVF, you see a sort of lumbar to cervical gradient where in the lumbar region with AVF, you get about 68 to 70% of cells being transduced, which drops down to about 40% in the cervical region. And we noticed this was obviously a pilot study, so we're limited in what we can infer, but we did notice an upward trend in both the number of transduced cells in AVF relative to AV9, as well as the average staining density of each cell. To measure different cells being transduced as well as the density of these cells, we used an image analysis program called CTA. It's a machine-guided algorithm that looks for different shapes to identify cell types and counts. It's guided by, it's trained by, and then checked by a board-certified pathologist at every stage to make sure we're counting the right cells. Groups such as Jim Wilson's in the last few years have reported dorsal root ganglion toxicity in non-human primates following AAV administration, particularly at high doses. So we wanted to look at DRGs to see how AVF did with them. And we were surprised that despite the slightly higher expression in interneurons and motor neurons, expression in DRGs with AVF was actually lower than that of AV9, with staining intensities similar between them. We were surprised to see this. Also, when we looked in the sciatic nerve, we saw sparse but consistent transduction of Schwann cells with AVF that we have never seen before with AV9, which we were really interested to see. When we examined the same tissues using qPCR, we were surprised. Despite a trend of higher expression histologically, we actually saw fewer genomes per cell with AVF transduced samples than with AV9. We saw this in both the spinal cord as well as in peripheral tissues that we looked at, namely the liver, spleen, and heart. Looking at data of individual samples, you can see that the expression is quite variable, which to us highlights the need to perform a larger study, hopefully ideally with a range of doses. So to summarize this part of the talk, we took AVF and moved it from the mouse to the primate and saw really successful transduction of the spinal cord in macaques, even at a low dose. We saw a slightly higher trend of AVF expression compared to AV9, with, again, slightly more restricted expression, it seems, in the DRG. And these, again, are things we would like to elaborate further with larger N numbers in a bigger study. We're also interested in looking at things like expressing, or rather administering a higher dose, as well as looking at self-comp vectors to see if we can expand expression. Interestingly, looking at other papers in the literature, several have looked at self-complementary vector in the spinal cord using similar and higher doses to what we used with AVF, and we're getting similar levels of expression, despite the fact that we were using a single-stranded vector. So again, I think a self-comp vector approach could be interesting to explore. So when last we left AVS in this story, we saw that it drove high local transduction in the brain as well as in peripheral organs, but wasn't able to cross the blood-brain barrier. We were curious whether this high local transduction would translate to the inner ear. Now, transducing the inner ear with AAV isn't trivial, and the hair cells of the cochlea in particular can be difficult to target. Inner hair cells, many serotypes, wild type and modified, are able to target inner hair cells, but outer hair cells in particular are quite difficult. And for inherited forms of hearing loss, targeting both inner and outer hair cells for the most part is required to rescue functional hearing. So to begin with, we wanted to test AVS in mice, so we injected neonatal mice at P1 with AVS carrying CBA-GFP. We injected a dose of 3 by 10 to the 10 vector genomes through the round window membrane, as you can see in that diagram of the right. There are a number of different routes you can use to transduce the cochlea. We opted for the round window membrane. So we injected at P1, and at P6 we harvested the cochleas and took a look. And what we saw was really widespread expression in the inner ear, looking at hair cells here, which are counterstained with myosin 7A. we see almost total transduction of inner hair cells and still very high levels of transduction in outer hair cells. And you can see in the graph on the right, there's an apex to base gradient of transduction. This is very normal when you're dealing with hair cells in the cochlea. But still, we saw up to 80%, 70-80% of outer hair cells in the apex being transduced, as well as 50 or 60% in the base, which we were really happy with. And we looked at other cells in the cochlea besides hair cells, looking in the lateral wall, and Claudia, sulcus cells. These are supporting cells that are very important and implicated in deafnesses such as GJB2 related hearing loss. We saw very high levels of expression as well. On the right, you can also see transduction of vestibular hair cells in the macula that were, again, we saw very widespread, consistent expression. We used ABRs and DPOEs to examine if there was any hearing loss as a result of this expression, because it was very widespread, a relatively high dose. We saw no difference between transduced samples and non-injected ears. So overall in mouse, we saw really widespread, really consistent expression with AVS in pretty much every hair type of cell we looked at, in particular hair cells, as well as a lot of supporting cells. So as well as expression levels, we were curious whether AVS would be relevant in a clinical context. So we decided to test it in a mouse model of Usher syndrome type 3A. Usher 3A is caused by mutations in clarin-1 and actually has a relatively slow development of hearing loss, with deafness beginning in patients as late as the second decade of life, which gives you a window of opportunity to intervene. And so we used a mouse model that reflects this. Rather than a straight clarin knockout, we have clarin-1 being driven off an 801 promoter, which causes it expresses for about a week before petering out, and this model mimics human disease development really well. So we loaded AVS up with a transgene of clarin-1 being codon-optimized clarin-1 being driven off a CBA promoter, including both clarins 5' and 3' UTRs. And we found the AVS clarin vector provided really good rescue that lasted for the entire length of our study of 5 months. So if you look at the top row, the ABRs, you can see uninjected mice in green. And while they retain some level of low and middle frequency hearing at around P35, by 2 months or 3 months, mice are completely deaf. However, if you look at the purple bar, which is treated mice, you see at low and middle frequencies right up to 1622 kilohertz, we have wild type level hearing restored or maintained, as it were, in treated mice. The dotted black line indicates an equivalent wild type. So we were really pleased with this, and this is reflected in DPOEs as well, which are an indicator of outer hair cell function. We see again in low and middle frequencies, this corresponds to the apex and middle of the cochlea. We got, again, wild-type levels of rescue that were maintained throughout the lifetime of the study. And we see this reflected not just in hearing, but also in morphology. When we looked using SEM, whereas uninjected mice, there was a bit of P60, there's a bit of fall off. The morphology of the hair bundles on top of the hair cells are either missing in some cases or damaged, and by 5 months they're almost missing entirely, and what's left are quite disorganized. Even at 5 months in treated animals, we see very good bundle preservation, excellent bundle morphology, which reflects what we see in hearing preservation. So far we've been able to show that AVS does drive really excellent expression in the mouse inner ear, both when looking at GFP expression as well as protecting loss of hearing in an Usher 3A mouse model. Obviously, the next logical step was to see whether this would translate to larger animals, to a non-human primate. And so we embarked upon another small pilot study. We injected the same AVS CBA GFP construct into a small number of ears, into 4 cynomolgus macaque ears at a range of doses, from a low dose of 8 by 10 to the 10 vector genomes up to a higher dose of 5.8 by 10 to the 11 VG, along with a PBS injected sham control. The results we saw were very similar to those of the mouse. At high doses, at the 4 by 10 to the 11 vector genomes dose, we saw very high levels of expression throughout the organ of Cordy in hair cells as well as in supporting cells. In the lower dose, the 8 by 10 to the 10 ear, we saw much less expression in the hair cells in particular, which I'll show you in a second. But we still did see widespread expression in places like the lateral wall. Zooming in, looking at the highest dose ear from a different angle, again, we see wide transduction of hair cells, of supporting cells, and just, it's a sea of green, really. Looking at the organ of Cordy and looking at hair cells in particular, at the high dose we saw, just like in the mouse, almost total transduction of inner hair cells as well as very high levels of expression in outer hair cells. Right up to the middle of the cochlea we see complete transduction of the outer hair cells, and this falls off relatively sharply towards the base. In the lower dose ear, the 8 by 10 to the 10, we actually didn't see any significant gene expression in the hair cells. And this mimics previous work we've done with another vector, with PHPB. And just like in the mouse, as well as the hair cells, we were curious about AVS expression in supporting cells as well. And looking throughout the organ of Cordy in the lateral wall, in the spiral ligament, in Henson's Claudius cells, the sulcus epithelium, again, we see really consistent widespread expression of AVS at the higher doses. And as I said before, expression in the lateral wall in the low dose is still very high. We wanted to make sure that the vector was safe in the ears of these primates. So we did ABRs of 4 of the treated ears before and after, including the PBS injected ear. And you can see these ABRs, they are flipped compared to the previous data in the mouse, so lower on the graph, as it were, is worse. And for 3 of these ears, the PBS treated ear as well as the low dose and one of the high doses, we saw no difference before and after injection. However, with one of them, you can see that in the bottom right, the ear was completely deaf post-injection. The reasons for this aren't entirely clear. We're still not sure on it. We looked using H&E staining. We wanted to look for markers of inflammation or damage, and what we saw was really only mild focal immune infiltration that was the same in that ear compared to the other ears. There was the levels of GFP expression and the numbers of apparently viable cells was the same in this deaf ears and the other ears. So it remains to be seen, it remains to be established what caused this. The surgery involved in injecting these monkeys is quite difficult. It's quite long, it's several hours, and so there may have been some sort of issue with the surgery or with the recovery. We're not sure. There's nothing that we've seen so far to indicate that the vector or the GFP was the cause of this, though. So to summarize this last section, we think AVS is really an excellent preclinical candidate for treating inherited hearing loss in the inner ear. In both the mouse and the monkey, we see really high levels of expression in our Clarent3, our USH3A model, or Clarent1 rather. We see really good levels of protection at low and middle frequencies. And in non-human primates, once at our higher doses, we get very high levels of expression across the cochlea. We think the levels of transduction in supporting cells indicate that this could be useful for treating GJB2 related deafness, and the high levels of expression in the hair cells open this up possibly to treating a range of diseases. So with that, I'd just like to thank everyone involved in making these projects a reality. In particular, Casey McGuire and David Corey, as well as Marina Ivanchenko. With regard to the inner ear work with AAVs, none of this would have been possible without Marina. So thank you so much. And thank all of you again for listening. I'm happy to take any questions you have.
Thank you very much, Killian. That was a great presentation. So hello, everyone. My name is Maoyan Wen, and I am a Global Product Manager at GenScript, and I'm going to be moderating questions for Killian's talk. So we've got a few questions here, so let's see. The first question is, how well do AAVF and AAVS produce compared to AAV9?
That's a great question. Roughly speaking, AAVF produces a little bit worse than AAV9, probably about 2/3 as well, and AAVS produces as well as, from what we can see. Sometimes a little higher, sometimes a little lower.
Okay, and then we've got another question. Since AAVs don't normally integrate, do you know how long the AAV expression lasts in supporting cells and lateral wall populations?
That's a really good question, because as I was saying, targeting supporting cells is vital for treating diseases like GJB2 related deafness. And the fact is, as far as I'm aware, there's still no consensus on how readily supporting cells and other cells of that type actually divide. We haven't done very long-term studies ourselves in any animal to see how long they are. In our hands, the expression has certainly lasted to the sort of end point, in mice probably about 6 months. You certainly see expression that long, but long term, I'm not entirely sure. But it is a great question.
Thank you. So we've got another question, which is, were there any differences in formulation between AAVF and AAVS in the spinal cord versus the inner ear studies?
No, they were both just formulated in PBS identically.
Thank you. And we've got one last question. Did the expression of GFP from AAV9 / AAVF outside of the central nervous system come from leakage during delivery or some other mechanism?
That's something we're curious about as well. It's possible that with intrathecal delivery, some leakage does happen, but it's also we're not entirely sure. It could just come through normal processing through the CSF, or it could come through a surgical leakage. Both are possibilities, but we've seen that in other studies. It's pretty universal that if you inject intrathecally, you see peripheral expression as well.
So that's all the questions that I have. So thank you very much again, Killian, and thank you everybody for attending this session.
And thank you so much for listening to me talk.
All right, thank you.
Bye-bye.